So, good afternoon, everyone, and welcome to your subject in Type 22 Agricultural Biology. So, for this video, we're going to discuss the Unit 3 of your subject, which is genetics. Last time, please check the video for the last meeting that we have about self-reproduction. And now we're going to proceed with Unit 3, which is genetics. And we're going to start with Mendelian genetics. So it is actually called Mendelian genetics because it was he was the one who is the founder of genetics, and he from 1822 to 1884 he was the responsible for the laws governing inheritance of traits within his study. So Gregor Mendel is an Australian monk who studied the inheritance of traits in pea plants and developed the law of inheritance. Gregor Johann Mendel was born between 1856 and 1863 and cultivated and tested some 28,000 pea plants. He found that the plant's offspring retain traits of the parent. So this is the actual site of the Gregor Mendel's experimental garden in Czech Republic. So Mendel stated that physical traits are inherited as particles known as the particulate inheritance, although he did not know that the particles were actually genes. And there are various genetic terminology that needs to be defined. Like for instance, we have trait. It is any characteristic that can be passed from parent to offspring. Is heredity, and another one is heredity is the passing of traits from parent to offspring. So there are types of genetic process: the monohybrid cross, or the cross involving a single parent, like the flower color, and the hybrid cross involving two traits. So Punnett square is used to help solve genetic problems. So as you can see, this is the male gametes, the male parents, and the female gametes. And if you, in the Punnett square, it's just pairing them on the square as shown in the diagram. So the P, for the P, small p, and another small p from the female gametes is equal to the two small p's, same as the double capital P's and so on. So to make the Punnett square, this will allow geneticists to predict the possible genotypes and phenotypes of offspring. In this example, the parents are heterozygous for yellow P allele. Y is to Y. So this is the parent one, the heterozygous Y's and Y, and the parent two also is Y and Y. So to make the grid like this and place the alleles of the gametes of one parent along the top of the grid and those the other parent along the left side of the grid. And then you fill the grid, combine the parent alleles inside the boxes. The letters show the genotypes of the offspring. So when you pair two capital Y and then one from the female, the parent one capital and one small y. And in the second pairing is one capital Y and one small y. And the fourth pairing is one small y and plus another small y is 2 small y. So the genotype ratio is 1 is to 1 is to 1 is to 2 and is to 1. Meaning there is one double y combination and there is two um, big letter Y and small letter Y combination and one small letter Y combination. So using the law of dominance to determine the phenotypes and the phenotype ratio of the offspring. So you have here the phenotype ratio is 3:1, meaning there are 3 yellow peas to green peas. Since the capital Y stands for the yellow pea, the small y stands for the green pea. And because the capital P is more dominant, the capital Y, is a more dominant, so it will color yellow while there is only one green pea. So the designer genes, known as alleles, are 2 forms of the gene which can be dominant and recessive. The dominant is the stronger of 2 genes expressed in the hybrid, represented by capital letter R. So genotype is a gene combination for a trait like the double capital R, 1 capital R, and R, small r, and while the phenotype is the physical feature resulting from a gene combination. So the genotype and phenotype in flowers, for example, we have R is for red flower, the capital R, and the small r is for yellow flower. So all genes occur in pairs, so the 2 alleles affect characteristic possible combinations are, for genotype, there could be 1 double capital R, 1 capital R, and 1 small r or double small r. So the phenotypes for the capital R is red, the 2 capital R is red, and the second one, the first capital R with a small r is still red because it's dominant, and the color for the double small letter R is yellow. So talking about genotypes, it can be homozygous or heterozygous. I'm sorry for the slides. You can always go back and check this one later on. So because I cannot maybe if I open this one, then I can edit it in Google Slides. So I can show you the whole, the whole text in the slides. So while waiting, we've done this one. So another types of genotypes are homozygous and heterozygous. So homozygous are gene combination with 2 dominant or 2 recessive genes. So meaning there are double capital R or double single R, small letter R, and they are supposed to be pure. While the heterozygous genotype are genes with a combination of the small and the big letter R or dominant and recessive genes. So we are now here on the genotype. Okay, so in here, so the genes and in both genes and environment will determine the characteristic of a particular multicellular living organism, including human beings. So they say that 90% is genetic. The actual, identity of a person is 90% genetics and 10% environment. So all in all, that determines your qualities as a human being and as an organism, whether plants or animals. And we know that human beings belongs to the plant kingdom. So this is exactly what is the Mendel's pea plant experiments. So he used actually peas because, or the Pisum sativum, because it can be grown in a small area and produce lots of offspring, and also produce pure plants when allowed to self-pollinate in several generations. And they can also be artificially cross-pollinated. So, so I'm sorry for this, because some of the slides actually are not showing well. So the reproduction and flowering plants, it's involving actually, you have known here, this one in your biology lessons before, that this is the reproductive organ of the plant containing the stamen or the male reproductive organ, and the pistil and the ovary is the female reproductive organ. So the pollen contains sperm produced by the stamen. These are the pollen from the stamen, and the ovary contains the egg inside the flower, and then the pollen is carries the sperm to the eggs for self-fertilization, and then self-fertilization can occur in the same flower, and if it's not, it's therefore cross-fertilization can occur between flowers, especially for those are incomplete flower. Incomplete flower meaning they don't have the both male and either one of the male or the female organs. So just like the monoecious plants, they only have they have both separate male and female flowers. So they are cross-pollinated, or in some other cases too. So the Mendel's experimental methods use hand-pollinated flowers using the pen brush and using the paintbrush, and he could snip the stamens to prevent self-pollination, covered each flower with a bag of cloth like that, and he traced the threads through the several generations. So the Mendel produced pure strains by allowing plants to self-pollinate for several generations, so as shown on the figure. And then after that, there were actually 8 pea plant traits that was produced. In terms of seed shape, there were a round or wrinkled, which is represented by capital R, and wrinkled small letter R. In terms of color, yellow is capital Y and green is small y. And for pod shape, smooth is capital S and small letter S for wrinkled. And pod color, green for capital G and yellow for small g. And for the seed coat color, we have gray or capital G and white as and small g. And for the flower position, the axial is capital A and the terminal is small letter A. And for the plant height, capital letter T or short small letter T. And for flower color, we have purple, capital P or white small p. Now let's take a look with the combinations here. In pictures, these are the pea plant characters studied by Mendel. We have smooth, wrinkled, yellow and green, inflated and wrinkled, green and yellow. And we also have purple, white, and flower position on stem and at the tip, stem length, tall, and dwarf. So those are just some of the characteristics. So based on the experimental result, the ratios of dominant to recessive in Mendel's plants are, we have dominant trait are the small seed, yellow seed, inflated, green, purple flower, and on stem and tall stem, while the recessive traits were wrinkled, meaning they are the small letter R or Y. Green seed, wrinkled pod, yellow pod, white flower, flower tip, dwarf stem, and average or ratio of all traits is 3:1, meaning these are the dominant trait. They dominate. They always comes, you know, visible, while the recessive trait is not so visible in the combination. So the ratio of dominant to recessive in the second generation, filial generation, is actually already 2.96 is 2:96, 2.96 is to 1, which is 5,407 for smooth and 1,850 wrinkled or so on. So usually the ratio of the dominant 3:1 recessive. So did the observed ratio match the theoretical ratio? So the theoretical or expected ratio of the plants producing round or wrinkled seeds is 3:1 wrinkled. So Mendel's observed ratio was 2.96. So there is like 1:1, 2.96 is to 1. So the discrepancy is, well, maybe brought by statistical error. So the larger the sample, the more nearly the result approximate to the theoretical ratio. So it's actually, when you conduct experiment, it's very important to have a large, large population, so you can estimate of the standard error properly. Now we have the so-called generation gap. We have parental or P1 generation. This is the parental generation of breeding experiment, while the F1 generation is the first generation offspring in a breeding experiment, or the first filial generation from breeding individuals from the P1 generation. While the second or F2 generation is the second generation offspring in a breeding experiment, which is the second filial generation from breeding individuals from the F1 generation. So following the generation, so it starts with the parent generation and the first filial generation and the second filial generation. So when we cross the two pure plants like the dominant T and the recessive T, so they all result in hybrids with heterozygous dominant and recessive traits. So if another on the second generation, when we go cross the two hybrids, you get 3 tall and 1 short, as shown on here. So let's talk about monohybrid process. So monohybrid cross is, for example, the trait is a seed shape. So there are 2 alleles, brown and wrinkled, as represented by big letter R and small letter R. So when you cross the brown seeds with the wrinkled seeds, so this using the Punnett square, so you will pair the first big letter R with a small letter R. So the combination is big and small letter R. So the capital or the big letter should come first, despite of their arrangement. And pairing again is R and R. The second pairing is first letter combination of big letter R and small letter R. And the next combination is again for big letter R and small letter R. And the third combination is the big letter R and small letter R. So All in all, they actually have the same combination because you are crossing a monohybrid cross. So the genotype is all big letter R and small letter R, and the phenotype are all round since they start with they have all the capital or the big letter R, which is the round one and the dominant um, allele. So we have a genotype ratio as alike, all alike, and a phenotype ratio as all alike. So they are all actually round. So for the monohybrid, for a review of the monohybrid cross, homozygous dominant times homozygous recessive will result the offspring all heterozygous hybrids, while offspring and the offspring is called the first filial generation, and the genotypic and phenotypic ratio is all alike. Next, for the second filial monohybrid cross, using again, if you are going to cross the first filial monohybrid cross, meaning they have the same trait and alleles. So we have again combination of the round and wrinkled. So you will have one, the first pairing, 2 capital R, and the next pairing is between small and capital R, and then R in the small capital, big letter and small capital R, and the last pairing is both small letter R. So the genotype is we have first all capital R and combination of the capital R and small letter R, or the round and wrinkled and all wrinkled or small letter R. So the phenotype that will come out is round and wrinkled with a ratio of 1:2 and 1. While the phenotypic ratio is 3:1. It means there are 3 round ones and small wrinkled or 1 wrinkled seed shape. So as a review, in a heterozygous and with cross, the offspring would be 25% homozygous dominant R and RR and 50% heterozygous combination of big letter and small r, and 25% homozygous recessive trait of small letter R offspring for second filial generation. So the genotypic ratio is 1:2:1. And the phenotypic ratio is 3:1. So what do the peas look like? So as shown on the figure, some of these peas have a smooth texture while the others are wrinkled. Now, Mendel then crossed a pure and a hybrid from this second filial generation known as the F2 or test cross. So there were 2 possible test crosses, the homozygous dominant times the hybrid and the homozygous recessive times the hybrid. And say again, homozygous, they have the same dominant capital R, and homozygous recessive is the same small letter R. So in the F2 monohybrid cross with a seed shape again, so we have between round seeds and another round seed, but a heterozygous round seed. So when you pair them, so you will have two rounds, homozygous round seeds, and one heterozygous brown seed. So the genotype is both big capital R and combination of big capital and small letter R, but the phenotype is all brown because it starts with the dominant R, and the genotypic ratio is 1:1. Uh, yes, and the phenotypic ratio is all alike. So in the F2 monohybrid cross, or the second combination between wrinkled seeds and brown seeds, as shown here, so you will have two brown dominant seeds, heterozygous, and two homozygous wrinkled seeds. So with a ratio of 1:1, because there are two heterozygous brown and two homozygous wrinkled. Next, so therefore, homozygous cross between heterozygous hybrid will result into 50% homozygous R or brown or wrinkled, and 50% heterozygous brown and wrinkled. And the phenotypic ratio is 1:1, and it's called test cross because the same offspring have the same genotype as the parents. So you can practice your process, no, in your own worksheet. So you can work with the parent generation for F1 and F2 process for each of the pea plant traits that is. There were more. It's not only the example that I've given you is just the seed sheet. So you can have the others to be 7 pea plant traits for your practice. So and after this experiment, Mendel's have actually proposed the different laws. So there were the results of the monohybrid process. So inheritable factors or genes are responsible for all heritable characteristics, and phenotype is based on genotype, and each trait is based on 2 genes, one from the mother and the other from the father. True breeding individuals are homozygous, both alleles are the same. So the first law is the law of dominance. So in a cross of parents that are pure for contrasting traits, only one form of the trait will appear in the next generation. And all the offspring will be heterozygous and express only the dominant trait. So the dominant trait here is the brown seeds. No, this is the recessive as represented by small letter R. This will all yield in a heterozygous but all brown seeds because the letter R or capital big letter R here is dominant. So in the law of dominance, the 3 genotypes yield 2 phenotypes, the yellow and the green with all this combination. For another law that Mendel proposed is the law of segregation. During the formation of gametes, eggs or sperm, the 2 alleles responsible for a trait separate from each other. So the alleles for a trait are then recombined during the fertilization, producing the genotype for the traits of the offspring. So applying the law of segregation, the female parent with a combination of tall and short alleles. So with a 1:1 female gametes between parent with another male gametes with the mix or combination of the tall and short. So the phenotypic ratio here will become 3:1. It's because it is just how the segregation. It's because of applying the Punnett square that we have learned. This is in relation to law of segregation. So you have 2 tall and 1 short. So the law of independent assortment is alleles for different traits are distributed to sex cells and offspring and independently of one another. So this law can be illustrated using the dihybrid cross. So dihybrid cross is a breeding experiment that tracks the inheritance of the two traits. Then there's law of independent assortment. Each pair of alleles segregates independently during gamete formation with a formula of 2n is equal to n to the number of heterozygotes. So if, for example, how many gametes will be produced for the following allele or arrangement? So remember this formula, 2n is equal to the n is equal to the number of heterozygotes. So the answer would be, there will be, since there are two heterozygotes between R and Y, so there are 2 to the 4th, so 2 times 3 is 4 gametes. So we have the, if you use the Punnett square, you will have a combination of dominant R and Y, and dominant R and recessive Y, and recessive Y and dominant Y, and recessive R and recessive Y. This is for the dihybrid cross, meaning you are crossing two traits, different traits. So in the second combination with A, B, C, D, so these are all the combinations using the Punnett square. So there are actually three heterozygotes, 1, 2, and 3. So since this C is homozygous, so 2 times 2, 4 times 2, 8. So there are 8 gametes. And for M, 1, 2, 3, 4, 5, 6. So there, in this combination, there is 6 heterozygotes. So that's equivalent to 64 gametes. So in a dihybrid cross, if you're going to cross traits like seed shape and seed color, so with the following alleles, capital R for round, small r for wrinkled, capital Y for yellow, and small y for green. So you will have this combination for the, these are all the possible gamete combinations using the Punnett square. Both dominant R and Y, then the combination of the heterozygotes, dominant R and recessive Y, and recessive R, and dominant Y and recessive R and Y. And for this combination, you will have the same. So if you are going to use the Punnett square, so you just, pair this one and then pair this one with this one. So you will have the result. So the result would be this one. I'm sorry. So again, this is the combination of the dihybrid cross. As you can see here, when you pair both the dominant R and Y, so you will have the double R, capital R, and double capital Y, and so on. So it's just the same with as we have discussed earlier, but this is just the combination of traits na, and you know that the pea plant have 7 traits, so you can have the monohybrid, and this time you can have the dihybrid, which is involving 2 characteristics na. So the round and yellow dominant are, if you're going to count them, there are actually 9. 1, 2, 3, 4. 1, 2, 3, 4, 5, 6, 7, 8, 9. I mean, the round and yellow lang palang ang combination are all 9, while the round and green combination are 3, and the wrinkled yellow is also 3, and with so the phenotypic is 9 is to 3 is to 3 and is to 1, while the wrinkled ratio is in here is 1. So I'm sorry for this. So next, so this is actually what in the phenotypic, the phenotypic combination. It's just the same with the first slide here, but this is with photos of figures. So again, there are round and yellow. There are 9. So 1, 2, 3, 4, 5, 6, 7, 8, 9. And round and green is 3. 1, 2, 3. And the wrinkled and yellow is 3. And the wrinkled and green is 1. And so the phenotypic ratio is 9 is to 3 is to 3 is to 1. So it's more clear. So test cross or a mating between an individual of unknown genotype and homozygous recessive individual. Like, for example, you have the blue eyes and a lacking a known genotype times blue eyes, and CC is the straight hair, but this is lacking. So using the Punnett square, you are going to pair them again for the test cross. So this would be the possible result when you pair them. So we have the BBC and let's say C for this. So these are the possible combination. You just pair the B and C. So there will be 2 recessive B and 1 dominant C and recessive C, and also here. So to summarize the Mendel's law, we have first the law of dominance. So when you cross parents with tall and short, so there will be 100% tall, because that's for the dominant tall. No? Using the Punnett square, you can try to segregate them. So in the law of segregation naman, between heterozygous, kasi ito is homozygous, sa lahat siya tall. 100% tall. And for this, heterozygous tall, for the law of segregation, so there only be 75% tall and 25% tall, short. And you can test that one in the Punnett square. And for lastly, for the independence assortment, So when you have dihybrid cross between dominant and recessive alleles, so you will have brown and green cross between brown and green. So this will be the possible offspring. There will be 9/16 brown seeds and green peas, and 3/16 brown seeds and yellow pods, 3/16 wrinkled seeds and green pods, and 1/16 wrinkled seeds and yellow pods. So we also have another incomplete dominance and codominance. What are these? So incomplete dominance is when the first filial hybrids have an appearance somewhat in between the phenotypes of the two parental varieties. For example, the snapdragon's flower with a red dominant R and white recessive R. So you have here the red dominant as red flower and the recessive as white flower. So when you cross them, so this will be the incomplete dominance combination. So this will produce the first filial generation, and all of this are becomes pink heterozygous pink. This will just explain, supposedly you will have all red flower, but because these are combined already, so incomplete dominance may occur as pink heterozygous. So this can also occur here as shown on the figure, the incomplete dominance with the parental generation white, recessive white, and dominant red. So the first filial generation will be a pink and another white, and if there are cross again with another white, there can be a combination of the second filial generation. So this is another incomplete dominance wherein 1/4 red will occur among pink, 1/4 pink, 1/2 pink, and white. So these are the possible combination. So next, codominance is also if two alleles are expressed in multiple alleles in heterozygous individuals. This happens with blood type, no, not only for humans but also definitely for humans, but most animals. The they don't have actually. Well, what I know is it's not as certain as humans when you speak of blood type. So definitely for blood type of humans. So we have type A and type B with this IA and IB, and type AB with A and B, and type O with I. So in the codominance problem, for example, the homozygous male type B heterozygous is crossed with heterozygous female type A. So the female is type A, this one, with A and I, and this is heterozygous, and while the homozygous male, which is type B, both the B. So of course, when you combine one A, A and B, and then B and I, and A and B again, and B and I. So there is 1/2 A and B and 1/2 B and I. So another codominance problem is when you cross male type O with female type AB, so the combination would be using the Punnett square with the AI, 2AI, and 2BI, 1/2, 1/2. So if a boy has a blood type O and his sister has a blood type AB, what are the genotypes and phenotypes of their parents? So boy has a type O and crossed with a girl type AB. No? So the answer would be the parents would have a genotype of the, because when you cross the IA and IB, you have A and B. So the phenotypes is A and B, and the genotypes, because you have type A and type O, right? Yes, you have type AB and type O. So you have parent with 1A and 1B and also type O. So the genotypes would be 1AI and 1BI with the phenotypes of type A and so there are also sex-linked traits. So these are traits or genes located on the sex chromosome. So the sex chromosomes are actually X and Y, actually XX genotype for females and XY genotype for males. I even heard that that's why there are a lot of gays, or maybe the tendency of men to become gay because they have both the X and Y genotype. You know? But I just have heard of that. And many sex-linked traits carried on the X chromosome. So example is that, and for an eye color in fruit flies, the sex chromosomes for fruit-like eye color is the XX chromosome female and the XY chromosome for male, X and Y. So when you're going to cross the red-eyed male with a white-eyed female with XR and Y and XR and XR. So remember that the Y chromosome in male does not carry the traits and only the X chromosome, as here. Many sex-linked traits carried on the X chromosome. So if you're going to use the Punnett square again, the red-eyed male and the white-eyed female. So this would be the combination. There will be 50% red-eyed male, female, and 50% 1:1 white-eyed male. So female sometimes can also be genetically carrier. In a sex-linked trait like the hemophilia, women are carriers and men have the phenotype more often. So like starting from Queen Victoria and King Albert. So Queen Alice was the carrier, and then passed on to, and then King Louis, and this and then to Alexandria is also the carrier, and then when it's passed to King Alexis, so he was the one who manifests the trait as hemophilia. So when we say hemophilia is actually so when we say hemophilia, this is genetics of the FAC gene. But before that, I've here some genetic problem solutions. So there is just a mix up of my presentation. So before we proceed with the hemophilia trait, so you can try genetic practice problems between a tall and dwarf pea plants with a P1 generation. No? We have discussed this from the previous lessons, but as your practice, you can do this. So breeding also the philia generation between heterozygous tall, and so you have here the solution, and So this is now the FHC gene. Hemophilia is correlated to the defects in the FHC gene. It's also found in the X chromosome. So in terms of inheritance, the hemophilia is an X-linked recessive disorder that affects only 1 in 1,000 males and very few females, since females have 2 X chromosomes and they are often the carriers of the genes. So hemophilia is when persons who inherit the FHC gene, they lack a clotting factor to prevent bleeding. And even simple cuts or wounds may be a serious problem for these people because they need substance or some injection in order to stop the bleeding. Or if the bleeding don't stop, then they die because of bleeding. And famous pedigree of hemophiliacs and carriers, as I have just showed you a while ago, is actually from Queen Victoria and the royal family of Russia. And the first daughter and the second daughter become the carrier, while the recessive gene passed from Russian family to Germans. That was what happened. So these were the Russian royal family. So again, you can try to solve this, to practice this genetic problems. And if you have questions and more clarification on this video or on this topic, you just feel free to comment, and you can always chat me in our group chat. So actually, in real life, my father also is diagnosed with polycystic kidney. So it's a genetic disease wherein also, just like hemophilia or FHC genes, the female, thank God, is just a carrier, while the male are the one which manifests. The so my father has a polycystic kidney. So we are 6 children of him, and we have 4 daughters. I have 4 sisters and 2 brothers, and hopefully they won't. We don't know if it depends though. If they don't have yet the polycystic kidney, but hopefully not. So thank you very much for listening, and I will see you again on the next video.